Hello, good afternoon, everyone. It's nice to see that I already have two students waiting for me here. Hi, Edmund, Soon, and Yu Xuan. Okay, nice to see you. These are my students. I'm very happy that you joined me today. So I'm also very happy that you want to learn more. Okay, so as long as you want to learn, very sure that you will do very well. Okay, the first step to improve yourself is to know that you want to do something about it, and I'm very sure that you will be able to do very well. Okay, so today we're going to have I'm going to finish off chapter 5 actually. Okay, so I've prepared some slides and I would like to share with you. And just do remember that today's class, this is a link that is always available, so you can always go to click on it again to watch it again and again. Okay, you can even share the link to your friends. Okay, so this is a DLP version, the English version. Just for the interest of those who are in taking your biology in BM, I'm having a same class, which is on Monday night at 9 p.m. But it will be including chapter 5.1, 5.2, and 5.3. That means the entire chapter 5. I'm going to do another class in BM version on Monday, the coming Monday. That means 24th of June. Okay, I'm going to share screen now. All right, I hope you can see this. Edmund and Yushan, can you see this? Okay, just type yes, teacher, I can see it. Oh, yes. All right, just to let me know that everything's okay. All right, are you looking at this? Okay, so this is the slide. Actually, what you're looking at here is a diagram showing the model of enzyme. Okay, so enzyme, just remember, enzyme is it's a molecular structure. Okay, it is a 3D. So it actually consists of, most of the enzymes are protein. So protein, as you know, is long chain, and they are folded into this three-dimensional shape. We call 3D shape. Okay, right. Good afternoon, Kong Kai Wen. Okay, nice to know you're joining my class. All right, so I'm going to start sharing now. I'm going to start my lesson. I've done 5.1 about 5 weeks ago, which is sometime in May. So if you go to my channel, right, you will be able to see all my videos there. Okay, so here, 5.2, let me start with, okay, now what is an enzyme? Okay, an enzyme is actually a molecule, right? But what is more important about this molecule is, this is a molecule that can only be made in a cell, right? It's an organic catalyst. Now, there are two words here. One, organic, means an organic substance is a substance that is produced by a cell or produced by a living organism. That means if the structure or the substance is organic, it is not found anywhere else, like for example in the air or in the earth, in the soil. It is not inorganic. For example, minerals are inorganic. It is already in the earth when the earth was formed millions of years ago, okay, when the earth started. So, but organic is substance that is only made by living cell. Example, protein, okay, carbohydrate, fats, oil, lipids, enzyme. All these can only be manufactured or synthesized by a cell. All right. And then the second word here, catalyst. Catalyst means something, a substance that can speed up a chemical reaction. Okay. Now we say mostly these enzymes are protein, mostly I would say, but there are certain enzymes which are later they are found. The example will be ribozyme. It is not an enzyme. It's actually a nucleic acid. Alright, okay, Cikgu Fanida. Yeah, thank you for joining my class. Terima kasih atas sokongan ya. Okay, so tolong sharekan pada murid-murid di Cikgu ya yang ambil biology. Saya ada kelas hari Isnin depan ya for the BM version. Okay, you can maybe tell your students. Okay, or you can share to your previous students. Okay, right. It is produced by living organisms, so that is meant by organic. Alright, okay, next. Now you will sometimes see this kind of molecules. Whenever you see this shows polypeptide chains, which are long and they are highly folded, and it is actually, there are hydrogen bonds that hold this shape together. So you can see, it's just imagine you have a long piece of, what do you call that? Look at my hand here. This is a yarn, okay? So this yarn here is like the polypeptide chain. And then you roll it up together, you get a ball, isn't it? You get a ball here. And how it maintains the shape is there is hydrogen bonds in between. So you see all this, this is the polypeptide chain that is all rolled up into a ball, right? Onto a shape, 3D structure. So this is an example, a model that shows your enzyme molecule. All right, so you also see diagrams like this. So anytime you see a diagram like this, it mostly represents an enzyme molecule. All right, it is a polypeptide. Most of them lah. All right, I wouldn't say all lah. Okay, let's start with, let's continue on. Now, let's look at the terms. When you talk about enzyme, you will have this another term called substrate. Okay, what is substrate? Substrate is the substance that will bind, okay, or will be reacted upon by the enzyme. So the enzyme is to speed up certain chemical reactions. For example, A plus B, all right? So A is going to combine with B, but your enzyme works to make the reaction faster. So A and B is the substrate, okay? Enzyme is the pembantu. Enzyme is something that helps to speed up the reaction between A and B, okay? So the substrate is molecules as well. So you look all these pictures. This is very common pictures you will see when you look at, when you learn about chapter 5. So you will see this substrate 1, substrate 2. Now this A and B, or substrate 1, substrate 2, they are going to be joined together. So you'll see the green color thing at the product. All right. So the enzyme is the pink color molecule there, which is your substrate 1 will fit in, your substrate 2 will fit in, and somehow they will be joined together. All right. Now, this is another diagram on the left-hand side here. So, substrates bind. Bind means ikat or attach itself with enzymes at a specific site. There is a special place here where the substrate will join and attach onto the enzyme. So, that place is called the active site. S-I-T-E. Please remember, don't put S-I-D-E. S-I-D-E to tepi, okay? Tepi at the site. This is S-I-T-E, which means tapak. Tapak means the specific place. Okay, once it forms there, the entire structure is now given another name. This is called enzyme-substrate complex. Okay, remember, enzyme-substrate complex. You cannot change the word and say substrate-enzyme complex. It is wrong. Okay, we can't change all these terms. It's always called enzyme-substrate complex. So the entire thing now is called enzyme-substrate complex. Okay, and after that, you will see the reaction happens between your, whatever substance. Either is to make or to break. Either it's a catabolic reaction or anabolic reaction. That's 5.1. So if it breaks apart, right, you will see the products. And the products will leave the active site. Okay, same thing. When the product is joined together, you will also leave the active site, making way for another substrate to go in. Okay, to continue, to carry out the reaction. Okay, now let's look at nomenclature. Nomenclature is actually naming, the system of naming, giving names to the enzyme. All right. Okay, another thing is, students, if you're new to my class, you are always welcome to type in questions. All right, I will check your comment box from time to time and see whether I can handle your question. Okay, if I cannot handle your question on the spot, I will make sure that I give you my link, I give you my Telegram name. You can PM me, and then we can communicate through PM. Okay, so nomenclature is the system of naming. So it's very easy to identify your enzyme. Okay, your enzymes will always end, not always most of them will end with this word called ASE. Okay, so the naming of enzyme is based on the substrate or based on the molecule or the substance that is going to catalyze, is going to react upon. Okay, it's going to act upon. So let's look at example. If the substrate is maltose, okay, can you guess what is the enzyme that will act upon maltose? Can anyone guess? Okay, Matthew Lai, hello. Yeah, nice to see you here. Okay, can anyone guess? Well, look at we retain the first name, but we take away the OSE and we replace with ASE. Okay, so it's very easy to know your enzyme and your substrate because they are very close together. Okay, they are very similar. Okay, so maltose will be hydrolyzed by, will be catalyzed by mal. You just change your OSE to ASE, so you call it maltase. All right, maltase. I'm sure my students know, just that they are very shy to put in your answer here. Okay, I'm very sure you know. Okay, lactose will be hydrolyzed by, very easy, take over the OSE, add in the ASE, lactase. Sucrose, sucrase. So sucrase will react or act upon your sucrose. will catalyze the sucrose. Amylose is actually starch. Okay, so amylose will be, the enzyme for amylose is amylase. That means we say amylase hydrolyzes starch. Okay, starch is called kanji in the end. All right, okay, then next you have protein. Protein, you take away the IN and put in ASE, so you get protease. Protease is the general name for all the enzymes that catalyze protein molecules. Okay, there are many, many different forms of protein molecules. Okay, lipid, lipase. All right, don't forget your E, others become lipas. Lipas is the cockroach. Okay, then cellulose, cellulase. Okay, now you will see that most, in fact, more than half of all the names that you know, enzymes, they always end in ASE. This is the modern system, the modern naming system. But previously, the scientists have found there are other enzymes, but they don't follow this name because this system has not been invented yet. So you have things like called pepsin, trypsin, rennin. They are also enzyme. Okay, it's just that all these names, pepsin, trypsin, and rennin, they have been discovered. Okay, they have been used before they changed this system, before they introduced this system of ASE. So in order not to confuse everybody, so they keep on using the name of pepsin. They don't change it to pepsase. They don't change it to trypsase. Okay, they keep it as it is with pepsin, trypsin, and rennin. All right, okay, so that is the nomenclature, the system penamaan of enzyme. Okay, now we come to a very important part of the lesson. This is always popular question in your SPM, which is they always ask you what are the characteristics of enzyme. Whenever they ask questions, okay, they always base it on describe, okay, or say what you know about the characteristics of enzyme. Okay, so look at all these. This is in your textbook. I have put them here. I will explain one by one now. All right. So as you know, enzymes are biological catalysts. Catalyst means the function is to speed up biochemical reactions, the reactions that happen inside the cell, okay, inside a living organism. All right. Okay. So next, enzymes act rapidly. So they are able to catalyze. That means they are able to speed up the reaction of chemicals. Okay, and they also act rapidly, and they also can be reused actually. Okay. Now enzymes are required in small quantities. You don't need to put a lot. Okay, the ratio of enzymes to the substrate is much higher. Okay, there's a certain reason why we don't need so much enzyme. Okay, because the reason here is it is reusable. Okay, the enzyme, once it catalyzes the reaction of substrate, this product will leave the active site and another substrate will come in. So you can use the same enzyme again and again. Okay, the enzyme does not get destroyed. The enzyme is not altered in any way. It's not changed. So you can use it again and again and again. All right, that's why you don't need a lot of enzymes. Okay, next characteristic, the structure remains unchanged. This is what it means by that. That's why you do not need to have a lot of enzyme. They remain unaltered, unchanged, undestroyed. Also can use that word after a reaction. Okay, next. The reaction of enzyme are specific. Okay, this is very important. You don't have one enzyme that can catalyze everything. No such thing. Okay, if substance A, you need enzyme A. Substance B, you need enzyme B. Okay, your substance C, you cannot catalyze by enzyme A. You must have enzyme C. So it's specific, only this specific substrate, because of the shape, we call the active site. Later I'll show you the diagram. The shape, the configuration, the substrate will only fit, particular substrate will only fit the particular enzyme. Okay, there's no one enzyme that can fit all the substrates. Okay, they complement, the shape complements. Okay, next, most of the enzymes are catalyzed by, most of the reactions catalyzed by enzymes are reversible. For example, what do you mean by reversible? Okay, A plus B, you get, let's say, C. The product is C, right? You use enzyme X, let's say. A plus B, you use enzyme X, you get C. Now, you can use back the same enzyme. Your C can also be broken down to become A and B with the same enzyme. So the reaction can go forward, it can also go backwards. okay, depending on the quantity lah, right? So that one you don't need to know. As long as you know the enzyme can catalyze reaction, okay, forward and backwards, that means it is reversible. All right, and then enzyme activity can be slowed down. Okay, for example, if you put certain poisons, why are they called poisons? Because actually they stop the action of the enzyme. We call it inhibitors. These molecules or substances are called inhibitors. When this is present in the system, this molecule or substance, we call inhibitors, will stop this substrate from combining or binding to the active site. So now your substrate cannot go in. Your substrate cannot go in, that means your substrate cannot be broken down, cannot be catalyzed, cannot be reacted by this enzyme. It will not have a product. Okay, so this is the action of the inhibitor. It blocks, it stops, or it slows down the enzyme from acting. Okay, next, lastly, now some enzymes require cofactors. Cofactors, it can be vitamins. Okay, that's why vitamins is important, you know. Why we need vitamins? Actually, you say vitamins is good for your good health, isn't it? To make sure all your systems run very well. It's because certain vitamins are vitamin A, vitamin E, and so on, all that. They need this, the enzyme need this vitamin to make the enzyme work better. So these are called cofactors. Vitamin, and some of them can also be ions, like magnesium, zinc. Okay, certain ions, they more or less, they attach to a specific place on the enzyme, makes the enzyme work better. So without these vitamins and ions, that's why you get certain metabolic processes cannot occur. That's why you get all this penyakit, all the diseases will come in. So with that vitamins, certain ions, your health, your better, you know, the well-being of the person is better because your processes in the body are functioning very well. Your enzymes can work beautifully. All right, okay, so that's what it means by cofactor. They need cofactor to work. All right, okay, let's carry on now. So no questions so far, I think, right? No questions so far. I meant to just say hi, right? That was some minutes ago. So let's go and now. Enzymes, there are two types. Okay, all the enzymes in your that's produced inside the cell, there are two types. So one is called intracellular, the other one is called extracellular. Okay, when you say intra, cellular means cell, lah, right? Intra means inside. So these intracellular enzymes are actually enzymes that are made, they are synthesized, they are produced inside the cell, definitely. All enzymes are produced inside the cell, but it's also used by the cell. This enzyme is used, produced inside the cell and also used by that specific cell. They use it sendiri pakai. Okay, they use the enzyme themselves, the cell that produces it. Well, on the other hand, extracellular enzyme, these kind of enzymes, they are secreted, they are produced inside a cell, of course. All enzymes are produced by a cell, but they are secreted outside the cell. They are not used by this cell. Okay, so these examples will be digestive enzymes. The pepsin, the lipase, the amylase, that is used in the digestion of your food, in the stomach, in the intestine, and so on. They are all extracellular enzymes. It is produced by certain cells, which are called the glands that produces digestive juices. Okay, so what about intracellular enzyme? Another fact is, where are they? Which part of the cell produces intracellular enzyme? They are synthesized. They are produced by free ribosomes. Free doesn't mean per tumor. Free means these enzymes these ribosomes are not attached to your RER, not attached to your rough endoplasmic reticulum. Okay, you've learned in chapter 2. You have the structure called RER, which is, you know, connects to your nucleus, and on it you have a lot of these ribosomes that stuck onto it. So this is not the one that is produced. This is not the ribosome that produces intracellular enzyme. The ribosome that produces intracellular enzyme is not on the RER. They are floating in the cytoplasm. That's why you call it free ribosome. They are not attached to it. Okay, and the other one, the extracellular enzyme, these are the one that's produced by ribosome. Okay, later we will see the production of extracellular enzyme. Okay, so if you can answer that entire thing, can be a sub-essay question on its own. It carries about up to 8 marks. Okay, but it's very easy to remember because it's all facts. Okay, give examples, at least 1 or 2 examples from every type. Intracellular enzyme, remember, hexokinase and oxidoreductase. This is the enzyme that is used during cellular respiration to break down glucose to get energy for the cell. Okay, hexokinase breaks down the sugar. Okay, hexo is for the shape of the sugar, which is hexagonal shape, right? Hexagon. Next example of extracellular enzyme are all these digestive juices, digestive enzymes, amylase, pepsin, trypsin, okay, lipase, all the sucrase, maltase, and so on. All right, okay, this is very simple, very easy. Now let's go to the next part. Let's explain or describe the production of extracellular enzyme. Okay, how does the cell make extracellular enzyme, and how does it get secreted or produced or released outside the cell? As you know, extracellular enzyme is the enzyme that is released outside. It is not used by the cell itself. Okay, now number one, the formula. Okay, if you want to make something, you must have recipe. You want to make a certain dish, you have a recipe. Okay, you need the raw materials. So the recipe, or we call the blueprint, on how to make the enzyme or how to make the protein, it is inside the DNA. So in the DNA, right, the DNA is here, right? You have this, let me draw red color, right? So you have this, remember your DNA, right? All the DNA is inside your nucleus. So inside the nucleus, inside the DNA is this information to make your specific enzyme. So the information is there, all encoded in your A, C, T, and G. Remember your nitrogenous bases? This one, chapter 4. Okay, all these are your nitrogenous bases. So the code is there, the codon, to make your protein. Okay, and after that, how do you get this information out? Because the place to produce your protein is not inside the nucleus. It's actually outside. It's actually on the ribosome. So this formula, how to take it out, how to copy, how to bring out this recipe and give it to the ribosome. So you need mRNA actually. So the mRNA will come out, okay, it will be passed on to the ribosome. So the ribosome is the site of protein synthesis. Where you make a protein, the place is ribosome. The information is passed out through the mRNA. The mRNA copies the information from the DNA and pass it to the ribosome. Okay? Now, so this information is on the ribosome now. So the ribosome is now making protein. All right? So remember, it's protein, not yet enzyme. Because it's belum habis. The process of producing the enzyme is not finished yet. We don't call it enzyme. It's just protein yet. The protein first. So once this protein is made, it will be transported in the RER. That means it will come and jalan, move all around here. Okay, you see all these in my green lines here. These are the protein moving in your RER. Then once it comes to the edge means the tepi, the edge. What happens is, number 3, you will find little, what they call that, vesicles will butt off. It will pinch off from the RER, and it will hold this enzyme, protein, not enzyme. It will contain the protein. It will butt off from the RER. Okay, now where is it going to go? So these are called transport vesicles that carry protein. All right, that carry protein, which you cannot call it enzyme yet, not yet finished. Belum habis, it's only halfway through the production. So it has to go to another kilang, another factory. Okay, the other factory is called this one. Now, this is the Golgi apparatus. Okay, this is called the Golgi apparatus, which is going to further process the protein to become enzyme. So this vesicle, okay, let me draw the vesicle, right? This vesicle containing what? Containing the protein will fuse with the Golgi apparatus. When I put GA means Golgi apparatus, and pour the contents inside there. Inside there, things are added in. Glycogen added into the protein, you get glycoprotein. Substances are added inside so that you will get your enzyme later. All right. So this process, right, the transport vesicle now containing the protein will fuse with join up with the Golgi apparatus. And then what happens inside there? In the Golgi apparatus, the proteins are modified. Again, look at this word, modified. You can use the word repackage, modified, process, also can. These three words you can use. So now it's made into protein already. Into enzyme. And once this is the finished product, how is it going to get released out of the cell? So this is going to move to the edge. Okay, at the edge here, then it's going to butt off a little bit. It's going to pinch off, like you're playing plasticine like that. I just pinch off a little bit. You get another vesicle called, now you call it secretory vesicle. Okay, you don't call it transport. Transport is before. Transport is before your Golgi apparatus. After the Golgi apparatus, we call it secretory vesicles. And now they contain enzyme. You can call it enzyme already. Okay, I think I have a comment here. No comment. This is a O and a dash. All right. Lion tried. I don't know how to pronounce the name. Self-proclaimed meditative. LOL. Okay. So this is your secretory vesicle containing the enzyme. So what happens now? How to bring it out? This secretory vesicle will move towards the plasma membrane and it will fuse with the plasma membrane to secrete the enzyme out of the cell. So now you can call it enzyme. Okay, beginning is protein. Once it passes through your Golgi apparatus, it has been modified into enzyme and now it's released out of the cell to be used out of the cell. Okay, so this can be entire essay question, which will fetch as much as 8 marks, depending on the teacher, whether they want to give you 5 marks or 6 marks or 7 or 8, you see, depending on the points here. So you write a perfect essay. If all the points there, you can get full marks. Okay, no worries about it. This is nothing about it is all straightforward. Just need to remember. All right, okay, let's go to the next part. Now, mechanism of enzyme action. How do enzymes work? How do you explain, all right? How does it work actually? Okay, so you will see a lot of diagrams like this. You will have a big molecule here with a special shape there, and that part is a special, the indentations, the inversion and all that, that's called the active site. And you have the substrate that is going to bind to that place. Okay, so remember these three words: substrate, enzyme, and active site. And also one more, the product. Product is the hasil. What happens at the end of it, you're going to get your product. Okay, the substrate is like your raw material, the sebelum, before. Then you have the process, everything done, then you get your product. Okay, so enzymes are proteins. I mentioned mostly are protein. They are made up of polypeptide chains that are folded into 3-dimensional structure. This one I explained earlier. It's a 3D structure. It's not flat. All right. So it has an active site with specific configuration. Configuration is bentuk, shape, that will complement, that will match a specific substrate molecule, the shape of it. So only one enzyme will catalyze one particular substrate. Okay, because different substrate will have different shapes. Okay, so you must match the enzyme active site shape. Okay, so this is a substrate, and that is your enzyme. All right, after that you will get your product. Now the binding of a molecule substrate with the enzyme is specific. I use the word specific. It means you cannot have one enzyme that binds everything. You have this A to A, B to B, and so on. So we call this the lock and key hypothesis. Okay, now how to remember? A lot of students ask me, teacher, how to remember which is the lock, which is the key? Okay, you cannot interchange. A lock, which is the lock, which is the key, must be fixed. For example, I give you a simple way to remember LE. Okay, enzyme is the lock, substrate is the key. Okay, so how to remember LEKS? LE, remember your L stands for lock, your E is enzyme. So your enzyme is your lock. Okay, now K and S. S stands for substrate, K is the key. So your substrate is the key. Okay, so remember LEKS and you won't have it terbalik. So you remember your enzyme is the lock, your substrate is the key. You cannot put it the other way around. Okay, all right. Now, so let's diagram form to make you understand. All right, you got one lock, only one key can fit into it. All right, so you get your lock and key complex. So if you have your enzyme, same thing. This is enzyme with active site, and this is the substrate. You see the shape will match. We call it complement, and you call it this entire thing already fit inside there. You call it enzyme substrate complex. Okay. So you see all these diagrams, get used to it. Okay, you must know how to label where is the substrate, where is the active site, which one is the enzyme substrate complex, and which one is the product. The one on the left is catabolic reaction, breaking down. You see the substrate is bigger. Then your product is 2 units broken down to smaller units. But the one on the right hand side, this is anabolic reaction, 2 substrates, smaller molecule. Then you join it together, you get a more complex molecule. So this is called a anabolic reaction. Okay, so you need enzyme to catalyze it. Okay, let's carry on now. Now, how do enzymes speed up chemical reactions? How do they work? What does the enzyme do to make the chemical reaction happen faster? What does it actually do? Okay, so let's look at what it actually does. Okay, first thing, now, before any reaction can take place, okay, for example, you have A molecule and B molecule. They need to collide. They need to combine. I mean, they need to meet each other, right? So you have to have energy. It has to have sufficient energy in order for your 2 molecules to collide and then to join up together. So before a chemical reaction can take place, it must have enough energy. It must have the required minimum amount of energy. So imagine if you have 2 molecules, they don't move, they don't have enough energy. That means they don't move around, they don't meet each other. Your A is not going to combine with B because they will be separated, isn't it? So you need to have that enough energy in order for them to collide, meet up, and then they can join up. So this minimum amount of energy that is needed is called activation energy. Okay, so activation energy is the energy that is needed to break the bond, because in any chemical reaction, you have to break the existing bond and then make new bonds. Okay, so this one A, maybe inside the molecules have to pecah, then you will have to combine with the molecule B, make new bonds. So you have to break the bond first and make new bonds. Then you have the new product. Okay, so the activation energy is the energy that's needed to break the bond in the substrate before the reaction can occur. So if you look at this diagram, all right, your substrate energy level is here. Okay, in the beginning it's here. Now, before it can form the product, you need to supply energy. So it has to achieve the certain threshold of energy before you can get your product being produced. So it has to collect, absorb enough energy until it reaches the threshold. Then only the molecules will start to react. Okay, so you can see that without enzyme, I see quite high, the energy that you need to supply is this much. The energy that you need, this molecule, the substrate, needs to collect, needs to absorb this much energy before it can produce the product. Okay, this is the activation energy. So this level is called the activation energy, the amount there. Okay, next. So what do enzymes do? Okay, this level, this amount is called the activation energy. So what do enzymes do actually? Now, if you add enzyme into the system, right, your molecules, your substrate is going to, react, right? You add enzyme into the system. What happens is enzymes will lower the activation energy. That means your substrate do not need to absorb so much energy before it can start to react. Okay, now the rate, because you don't need to absorb so much energy because it takes time. You need to absorb more energy, it takes time. It'll take longer. You need to absorb, serap enough energy, then only start the reaction. But because it lowers the activation energy, the energy needed is less, you can start your reaction faster. Okay, the rate of biochemical reaction in the cell is accelerated. Accelerated means speeded up, dipercepatkan. So let's see the change in the diagram. Without catalyst, that means the picture on the left here, no enzyme, you find the substrate need to collect, absorb so much of energy before it can start to react. But what happens if you put enzyme? You add in enzyme here, all right? The second diagram here shows if you add enzyme, you see the energy level that is needed is only here. If not, you need this much. This is the activation energy without enzyme. And this one is the activation energy with enzyme. That one without. Okay, so now you don't need to absorb so much energy before the reaction can start. So therefore, we say enzymes function by lowering the activation energy. This is how we should answer the question. Okay, lowering the activation energy. So it will start the reaction faster. So you can see the difference in the level. All right. Without enzyme, you need to absorb more energy. Then only can start reaction. With the enzyme present, you also need absorb energy. Yes, you need to, but less. So if you less, faster. You don't need to wait so long. Okay, understand? Yeah. All right. Now next one. What are the factors that affect how the enzyme reacts? I mean, fast or slow. All right. There are 4 factors. So always remember these 4 factors. So I'll show you first temperature. Okay. So enzymes are sensitive to temperature. Very sensitive. Only certain the enzyme can react within a certain range of temperature. Then pH value. Certain enzymes will react better in different environment, different condition, acidic, alkaline, or neutral. Then substrate concentration, how much concentration you put in. Okay, the more you put in, of course, the reaction will be faster. And then another one is enzyme concentration. The more enzyme you put in, of course, the reaction will be faster because got more active site. Okay, so now we look at these 4 factors one by one. Okay, for temperature. All right, now temperature, this is a very popular question. Okay, why do enzymes only react at a specific temperature? Means the optimal reaction. Why at certain temperature it will be like enzyme act so fast, and how come different temperature it doesn't work very well? Your enzymes are very slow, or rather the enzymes cannot work at all. Okay, so how to explain? Look at the graph now. Yeah. When the temperature rises, when you keep on adding the temperature, that means from 0 to 5, 10, 15, 20 degrees Celsius and so on, you will find that the rate of reaction will increase. Yes. Okay, what is the reason? How do you explain why when I increase the temperature, okay, of course it will stop at a certain level until 37 only. Why when you increase the temperature, why do the reaction happens faster? This is how you answer. Because the kinetic energy of the substrate and the enzyme molecule increases. Because don't forget, temperature is actually heat. Heat is a form of energy. You give more heat, it is actually the kinetic energy of the molecules will increase. So they have a chance to collide. They will start to bump into each other. This increases the frequency of effective collision. So when they collide, they will actually join together. The substrate can bind to the active site of the enzyme. Okay, so more frequency for them to meet, for them to collide, and therefore the reaction increases up. You will find that in the diagram, every 10 degrees increase. So increase 10 degrees, the rate of reaction roughly is double. Okay, for example, let's say it's 5. If you increase to 10 degrees Celsius, all right, it will become 10. Then you increase another, it becomes 20. You increase another 10 degrees Celsius, it will become 20 40. The rate of reaction, let's say. But doesn't go on forever until it reaches the optimal temperature. This is the maximum already. It will not go any further even though you increase the temperature. So this is your maximum rate of reaction. Only at 37. Okay, remember this is only for animals, body. If plant enzymes, it's not 37, because plant require a lower optimal temperature. The plants have a lower optimal temperature. It is less than 30. It is actually around 27. So for plant cells. For animal cells, right, animals, the optimal temperature is 37, and that's the reason why the body temperature of many organisms, mammals especially, is 37. The enzymes work perfectly at this temperature. Okay, now look at what happens after that. At optimal temperature of 37, the enzyme rate is maximum. Okay, look at the word that you use. Temperature, we say optimal. That means the best temperature. Optimal means the best. Okay, and then the reaction, whether fast or slow, you use the maximum rate. Okay, not optimal rate, not maximum temperature, but optimal temperature and maximum rate. Okay, use the correct word, otherwise it's wrong. We're very particular, all right? So after the optimal temperature is reached, means you already reached 37. If you keep on increasing the temperature, it will cause, on the other hand, you have a reduction. It doesn't go up any higher, but it will drop. Okay, you will find that it's not symmetrical. After 40, it drops very fast. So it's not a bukit, it's not symmetry. The left-hand side is a little bit more less steep. The one on the right-hand side is steeper. That means it drops quite fast. Especially at 50 or 60, it's almost like zero. Okay, now what is the reason? After 37, your enzymes don't work very well. The reason is because you have to say that the enzymes are denatured or they undergo denaturation. This is where the chemical bonds that hold the shape of the active site or hold the shape of the enzyme is broken. So once it's broken, the shape of your active site is no longer maintained. It opens up, just like your ball of yarn. That's what I told you just now. Let's say this is the ball of yarn here. This is the enzyme with the active site. So if the bond is broken, so your string, like these are buka already. So when it buka already, your active site is gone. Your substrate cannot bind to it. All right, okay. So the active site changes, already changed shape. Therefore, substrate cannot fit into the active site of the enzyme. This is how you answer in order to get good marks. Not good marks, very good marks. Okay, this is how you answer. You don't beat about the bush. You must know what words to use. All these words that I highlighted, these are the words that you must use. Okay, let's go to another. Before that, let's look at the shape. So the normal protein should be like this. Let's say, for example, the one over here and the one over there. Okay, let's see what happens if denaturation happens. When denaturation happens because of heat, the bonds that hold the shape together, so all the hydrogen bond, they'll be broken. So once it's broken, you see the whole thing unravels. Your polypeptide chain doesn't hold that 3D shape anymore. So now it's loss of biological activity. That means it doesn't function already. And look at the shape of the active site. It is all, change shape. So when it changes shape, this substrate cannot fit anymore. All right. So now, one thing important about denaturation by heat, it is irreversible. You cannot bring it backwards. You cannot go back to original. For example, you heat up the enzyme is destroyed, it undergoes denaturation. Let's see what happens if I cool back the enzyme. I bring it back to lower temperature. Will it act again? Actually, no. It is irreversible. You cannot change it back because it's gone, right? You cannot make back the bonds by cooling back the enzyme. It's irreversible. Okay, next, pH value. Okay, pH value, So here you must remember 3 important enzymes with the pH value, Okay, most enzymes are active at 68 lah. That means the neutral condition. Example is a salivary amylase, where the value is about 7. You look at the peak there, right? Exception, most are better at neutral, but some have specific, they prefer more acidic environment. For example, Pepsi. Optimal pH is 2, around 1.5 to 2.5. And trypsin, this is another enzyme that is under the category of protease. It breaks down protein. It has an optimal pH of 8.5, which is slightly alkaline. Okay, all right. So you just remember P, A, and T. Pepsin, amylase, trypsin. PAT. All right. One acidic chonto. One is alkaline amylase. Neutral. The other one is alkaline. All right. Next one. Now pH value. Why do change of pH value will make the enzyme not active? Okay. Now changes in pH value will change the charges, not change the changes. Look at this word. This is called charges. Because the charge, is different already. Look carefully at the diagram here. Ions are actually, positive ions actually is what gives the acidity. Things are acid because of the hydrogen ions. Okay, there's a question here. If chemical bonds are broken, can enzymes be reused? Chemical bonds, you must see what is broken by what? Nia, Nabila. If it's broken by heat, they are irreversible. You cannot make back the bonds. But I'm coming to it now for pH. It is reversible. If it's not too extreme change of pH, if you bring it back to, let's say pepsin, it's now alkaline. It doesn't work very well in alkaline. But let's say you add in acid into your solution. Your pepsin, the bonds, the ions, the hydrogen ions will go back to the site, and now it will function as normal. Okay, look at the diagram. I'll show you this. So look at the correct pH here on the left-hand side. Here. So here you have, here is negative, here is positive, right? Positive, negative. So it will fit lah, you know, opposites attract. So let's say if you put in too much acid, you add acid into your solution. Okay, now what happens when you add acidity is hydrogen ion. So everything now becomes positively charged. When both sides are positively charged, it will repel. Repel means it will push each other. Your substrate will not fit into the active site. Same thing if you add in alkaline. Alkaline is your hydroxide ions. Okay, everything now, the active site and the substrate now becomes alkaline. So they will also repel. That means it does not fit into the active site anymore. Okay, but if you add in acid into the alkaline, you neutralize it back, or you put it even more as bring it back into acidic, you will reverse back the charges. Okay, so originally, if you already change the charges, your enzyme substrate complex cannot be formed. All right, but if you reverse it, you restore the charges. You by putting in, you know, if it's alkaline, you put in acid law. If it's acidic, you put in alkaline law. If you reverse the charge, the active site is reversed. The active site is restored. You can use your enzyme back. Okay, that's why Nabila, you asked a very good question just now. So it's caused by what? Is it heat or caused by change in pH? Okay, however, right, if extreme change in pH, it will also cause denaturation, which sometimes it cannot come back. Right? If it's too extreme now, the change in the pH value. All right, okay. Let's go to the next one. Time is running. So substrate concentration. Now the third factor. Okay, now substrate concentration means you keep on adding more and more substrate. So if you keep on adding more and more substrate, you don't change your enzyme. Okay, let's say your enzyme concentration is your fixed variable. I have a specific experiment here. I fix my enzyme. Okay, I do many types of experiment. I add in, I keep on adding my substrate. My enzyme don't change. You cannot change both. Your experiment is out. You cannot get your what they call valid results. You will change both variables. You only change what you want to test, and this is the manipulated variable. I keep on adding substrate. Let's say, for example, in the beginning, right, you'll find that the more substrate you add, you see the graph, it goes up. When the substrate concentration increases, the rate of reaction will increase. Just like you see the graph going up. All right. However, okay, what's the reason first? What is the reason first? First of all, because you got more active collision. You add in more substrate, there are more chances for your enzyme to collide with your substrate. So more effective collision, therefore more enzyme substrate molecule can form. Okay, so it will rise, it will increase until it reaches the maximum level. It will not go on forever. It will not go on forever. It will stop. Okay, you will get a constant value, constant rate after this. There is a reason why. Okay, I'm going to explain to you why. Why you keep on adding your substrate, you are not going to increase the rate of reaction until no stop. Okay, the reason is here. Let's look at the diagram. I've got enzyme that is orange color thing there, and the substrate is that gray color thing there. Your enzyme and substrate will fit together, right? Okay, now let's say we do an experiment. My enzyme concentration, I fix it at 8. Okay, in my experiment, my enzyme, I've got 8 molecules of enzyme. Okay, and then I start my reaction by putting in 2 substrate only. For example, right? 2. Now, can you tell me, maybe Nabila, can you tell me what is the rate of reaction? How many molecules of substrate can be broken down here? I've got 2 substrate, I've got 8 enzyme. Is my answer 2 or 8? Nabila, Mia Nabila. Is my answer going to be rate of reaction? Is it going to be 2 or is it going to be 8? Can you tell me? Or maybe Matthew, or Kaiwen, or Yushan, Edmund. Yes, good. Mia, correct. Because you only got 2 substrate, isn't it? Only 1 substrate will fit into 1 enzyme. Because I've only got 2 substrates, so I can only get 2 substrate produced per minute. Right? Okay. Now what happens if I increase my substrate? Let's say I increase to 4. Now I add my substrate. Instead of having 2, I put 2 more. Now tell me what is the rate of reaction now? What's my rate of reaction now? Mia or Matthew? Or Yushen? Anybody? Now I got 4 substrate. What is my rate of reaction now? Per minute, let's say. How many substrate can take part in this reaction per minute? Yes, Mia, good. I got 4, right? So you get the idea, right? I got 4. I will have 4. All 4 can take part. In that 1 minute, per minute, all 4 can take part because I got 8 enzyme. Okay, I keep on adding lah. I keep on adding 6 now, let's say. What is my rate of reaction now? Okay, of course, definitely you say 6. Okay, because I've got 6 molecules now, I got 6, right? Now what if I add some more? Substrate, keep on adding. Another 2 more, I get 8, right? Now what's my rate of reaction? All the 8 can take part. All the 8 can take part in the same time. I will have 8 as my rate of reaction. Now what happens if I keep on adding my substrate concentration? I put extra 2 more. What is my rate of reaction now? Okay, now it's already 10 substrate. Okay, anyone want to answer? What is my rate of reaction now? I've got 8 molecules of enzyme. I've got 10 molecules of substrate. How many substrate can take part in this reaction per minute? Yes, good, Matthew. Only 8. How come not 10? How come not 10? 8 is correct. Okay, why not 10? Because my enzyme not enough, isn't it? I can put in some more. I can put in 12 substrate, but my enzyme is only 8 because it's 1 to 1. One at certain one time, only this enzyme can fit into that active site. So if I add in some more, pun tak berguna because only got 8 molecules of enzyme, isn't it? So at maximum level, that means already maximum, the rate is already maximum because all the active sites are saturated. This is how you answer. Saturated means sudah penuh, fully occupied. Yes, no more active site to bind to the substrate. Good, Matthew. Yeah, no more active site already because I limit my active site. I only got 8. I put in 1,000, so no use. I don't have 1,000 enzyme. I put in 1,000 substrate. I only got 8. I still get 8 unique. Okay, so the rate of reaction does not increase when the substrate concentration increases. So therefore, what is stopping it? Can you tell me what is the problem now? Why cannot go up further? What is the problem now? Who is the one that makes the problem now? The problem lies with where? Where is the problem? The problem is now not enough what? Not enough your enzyme. Yes, so we call it the limiting factor. The enzyme concentration becomes the limiting factor. Please use this word. Okay, limiting factor is the factor that stops the reaction from going further or increasing some more because you don't have enough enzyme. Okay, you understand this concept? Yeah, it's very simple. So I'm going to go through the last one, which is the enzyme concentration. The same concept. The only change is the reason. The reason is slightly changed. So here I change Catabolic. I change it. Now I keep on adding my enzyme, but the substrate is fixed. Okay, I fix the substrate. I cannot change both. I fix my substrate. I keep on adding my enzyme. What is going to happen to my rate of reaction? Okay, so the manipulated is the enzyme. The enzyme is added up, adding and added lah. Okay, so the reason now, the beginning, you find you have the graph going increasing. When the enzyme concentration increases, the rate of reaction also increases. Right? You can see now. What is the reason? The reason is you got more active site. The enzyme is the active site. You got more enzyme, means got more active site can take part in the reaction. Okay, more active sites are present for reaction. Therefore, your reaction rate will increase. But until also there's a maximum level. After that, there's no longer any increase in the rate of reaction. So you can see the graph also will level off. It will become flat. Okay, it will level off. Now, let's see why. Okay, diagram form again. This one you will still understand. If you understand just now, you understand this one. Now, I fix my substrate. Let's say my substrate is only 8. My substrate only 8. My enzyme, I give 2 enzymes in the beginning. All right, I only got 2 enzymes, but I got 8 substrate. So now my rate of reaction is 2, isn't it? Because I only got 2 and only 2 can take part now. So what happens if I increase my enzyme? I put 4. All right, and then my rate of reaction will be 4. Yes, because now 4 substrate, more active site, another 2 can go in. So you got 4. So continue on 6. I put on, I increase to 6. My rate of reaction is also 6 because I got 8 actually, yeah. So now I increase it to 8 molecules of 8 enzyme. My substrate is 8. All 8 can take part. My rate of reaction is 8. Okay, what happens if I keep on adding enzymes? Okay, I put another 2 more, I get 10. All right, what is my rate of reaction now? Matthew, what is my rate of reaction now? What's my rate of reaction now? Huh? I got 10 molecules of enzyme here. I got 8 molecules of this substrate. Yes, good, Mia. Yes. Because you see now, only one can fit one only. I don't have extra substrate, I only got 8 no. Okay, so I still get 8, right? I don't get any more extra. I keep on adding enzyme pun tak berguna because I only have got 8 substrate. So what is the problem now? The problem now is you do not have enough substrate. Okay, so the maximum rate is reached due to the limited amount of substrate. So now what is the limiting factor now? What is stopping it? Yes, you get it already. Yes, the enzyme concentration. You're missing the word there. Matthew, you need to put in the word concentration. So now the enzyme concentration is the limiting factor. It's the one that's stopping it. Hey, no, enzyme. Enzyme is not limiting. Matthew, it's not enzyme there. Because enzyme is you can increase. Whatever you can increase is not the limiting factor. Whatever that is fixed is the limiting factor. And yet, Matthew, so you see, when we increase the enzyme, the limiting factor is the other one. Remember, you increase the enzyme, whatever you increase is not the limiting factor. Limiting factor is the one that cannot be increased. That means your substrate. Substrate is the limiting factor. Okay, remember, okay, so substrate is the limiting factor. Okay, lastly, application enzymes. This is more of application examples. This is common objective, right? And also structure. Now, in everyday life, we actually encounter a lot of enzymes. All right. Now, they are widely used in commercial sectors when they produce certain products. And most of the enzymes are extracted from bacteria, and some are actually produced synthetically. That means man-made. Okay, we use it in our food processing industry, a lot of things, right? Even for products, processing paper, making cloth and everything. We use enzymes. Okay, we're going to see some examples. Now you have something called immobilized enzyme. This is the immobilized enzyme technology. So nowadays, the enzymes they are certain combined with something else, which is called the inert, means not active, or something that is insoluble, to make the enzyme more stable. So the enzyme is more stable. The enzyme will not break down. It's not so sensitive to heat changes, not so sensitive to pH changes and so on. It's more stable. So the purpose of immobilized enzymes is to increase the resistance of enzyme towards the change in factors. That means not so easily affected by pH, not so easily affected by temperature. So it's good, right? Because it's more stable. It does not get broken down easily. Okay, it does not get changed easily. So advantages of using immobilized enzymes. Okay, enzyme is more stable. Okay, not so easily affected by changes in the condition. Can be reused continuously and can be easily separated from the product. These are some of the advantages of using immobilized enzymes. Okay, let's do some examples. All right. So I'm coming to the end of my class already. I will just hang on for a few more minutes. What I've mentioned here is everything you need to know. Everything you need to know about this 5.2 to 5.3. Okay, you don't need to search further information. Okay, everything is here. So, dairy industry. This is the pembuatan all these products made from milk. We call it dairy. The industry, tenusu. Okay, for example, making cheese. Okay, you need to dissolve and remove the fat for cheese making. So use lipase because lipase breaks down oil, breaks down fat. Okay, and then how do you convert the protein, which is soluble, carcinogen, all right, convert it into solidify to become protein inside the cheese. It's solid, so we use renning. Coagulate means is to make it to solidify milk protein. Milk protein is the casein. Okay, then you have certain people who are lactose intolerant. Some people who cannot drink milk, After they drink milk, you see the stomach, very, very bloated, vomit, and all that. Some people are sensitive, they cannot drink milk. So they have to take out the lactose. The reason is because they cannot break down the lactose, the sugar. The sugar inside the milk is called lactose. They cannot produce lactase, so they cannot drink milk with lactose. Otherwise, the lactose will start to ferment inside your stomach. It's all bloated and uncomfortable. So they produce milk that doesn't have any lactose. We call it lactose-free milk. So the lactase is used to hydrolyze the lactose, to break it down. Okay, next. Okay, next one. Detergent industry. Industry pembuatan sabun. Sabun, detergent, the soap powder. So you have this, oil stains, you need lipase. So lipase is incorporated to your detergent. Okay, and then you have also amylase. Amylase is to break down starch. Starch that is inside the sauces, the gravy and all that lah. Okay, so you break down the starch inside the sauce lah. And also like blood stains, right? Protease is incorporated into your detergent to break down anything that is protein, right? Protein inside your stains, blood and saliva. Okay, next you have next one. A bit slower. Okay, food processing. Protease, you see all these fish without the skin. The skin is stuck quite tightly to the meat, If you cook it, then it can be separated. But if you don't cook it, it's very difficult to separate unless you slice it. Okay, you want to peel off the skin, it's very difficult to peel off. So it's to separate the fish skin from the flesh. And also we use protease to tenderize the meat, especially beef and mutton, which can be quite tough. Okay, quite tough to eat, a bit hard to chew. So you can soften it. Pectinase, this is to break down pectin. Pectin is a substance in the cell wall. So if you break down the pectin, all right, the juice that you produce is not so, not so murky, not so cloudy. Wait, I switch on the, I switch on the charger. Okay, it's not so cloudy, so you get clear juices in your fruit juice. Okay, next, zymase. Zymase is inside the yeast. So you need your yeast to make beer and to make wine. Is to convert sugar into ethanol. Ethanol is the alcohol. People get mabuk when you drink your wine and your beer, because you have your zymase enzyme to convert the sugar inside the juice, the grape juice and everything, to become alcohol. Okay, then amylase is to make syrup, making syrup. Okay, converting the starch to sugar using amylase. Okay, another example, getting all this seaweed, not seaweed, agar powder. It breaks down, the cellulase breaks down cellulose, so you can extract the agar from the seaweed. You can get your tai choi ko, your agar that can solidify. Okay, right, next one, last one, right? I promise my class is coming to the end, right? So now application of enzyme, another one is, you see what is this? These are all skin industry. We call it leather. Tanning. The industry to make leather goods is called tanning. And tannery is the kilang. The kilang that produces all the hasil kulit is called tannery. So tanning industry is to make all this barangan kulit right? So trypsin is used to remove hair from the animal hides. Hide means the skin, the leather. Leather making industry makes the leather softer. Remove hair, you're not going to pull off one by one, there'll be too many. So you go and put this trypsin, it will hydrolyze and remove the hair from the skin there. All right. Okay, so I think that, some more. Okay, I think one more. Right, one more. Okay, textile, making two more. Textile is kain, industry kain, making cloth, remove starch in the fabric, okay, using amylase. Okay, and also you also need to use cellulase to break down the fibers, okay, of the because you cotton is actually a plant product. You have cellulose in the cotton. All right. Okay, lastly, in making paper, the paper industry, see here, right? You need to remove the lignin. Lignin is a substance that makes the wood hard. You need to, sort of break down all the pulp there. You get all this mushy stuff, but there's lignin there. Lignin will make it very hard. So you have to break down the lignin by using ligninase to break down the lignin in the paper making industry. Okay, I say rolls of paper that's soft. All right, so that's the class I have for you today. So here, I will continue with another lesson. I'm not sure whether next week or the next week, depends on the time that I have to produce the slides. Okay, but for sure that Monday I'm having a BM version, 9 o'clock PM. All right. So this is my channel. Just go to YouTube and type my name here. You'll be able to see my scenario, call it playlist lah. So I'll give you my what they call the Telegram post, a Telegram name. If you want to contact me, if you ask me questions, I'm most happy to answer for you. All right. So I'll give you my Telegram name here. If any questions you want to ask me, I will try to answer. I'll try my best. Okay. All right. Thank you very much for watching. I hope to see you another time. If you have any questions, please do contact me. All right. Bye-bye, everyone. Have a good week and see you next time. All right. Bye-bye.